Hello everyone, I'm Li-Feng Gao from Department of Immunology, School of Basic Medical Science, Shandong University. Today, we would talk about immunocytes. You may ask how the host defends against pathogen infection, such as novel coronavirus. To make clear this question, I would introduce the components of immune system and their major functions. Today, we are going to learn about immunocytes. The immune system is the structural foundation of immune function and is composed of immune organs and immune tissues, immune cells, and immune molecules. Based on their functions, immune organs are divided into two groups, including the central immune organs and the peripheral immune organs. Central immune organs include the bone marrow and thymus, both of which provide the places for lymphocyte development, differentiation, and maturation. Peripheral lymphoid organs include lymph nodes, spleen, and skin mucosal-associated lymphoid tissues. Peripheral lymphoid organs provide a place where immune response usually occurs. The bone marrow is the site of generation of all circulating blood cells in adults, including immature lymphocytes. The bone marrow is also where B cells differentiate and mature. In addition, secondary humoral immune response occurs in the bone marrow. Thymus is the site of T-cell maturation. The thymus functions in immune regulation by secreting thymic hormones and cytokines, which help convert thymocytes into mature T cells. The thymus also performs barrier function. T cells are selected through a process of thymic education before being released into the peripheral lymphoid organs as mature lymphocytes. After positive selection, T cells acquire MHC restriction. Negative selection confers T cells the self-tolerance. Mature T cells are CD4 positive or CD8 positive T cells. All the cells that participate in the immune response are called immunocytes. Among them, T and B lymphocytes play key roles in the immune response. T and B lymphocytes can differentiate, proliferate, and cause specific immune response after they recognize specific antigens. Thus, they are called immunocompetent cells. Here, list the major immunocytes. Immunocytes include innate immunocytes and adaptive immunocytes. These cells belong to innate immunocytes. We just introduced NK cell, macrophage, and dendritic cell. NK cells are an important part of the early innate response to kill pathogen-infected cells and tumor cells. Human NK cells express CD16 and CD56. NK cell receptors include killer lectin-like receptor KLR and killer immunoglobulin-like receptor KIR. Killing by NK cells depends on the balance between activating and inhibitory signals. Activating receptors include NCR, NKG2D, and CD16. The ligands for activating receptor may include molecules that are commonly expressed on the surface of motor cells and some microbial products. The inhibitory receptors of NK cells bind to self-class 1 MHC molecules, which are expressed on most normal cells. When both activating and inhibitory receptors are engaged, the influence of the inhibitory receptors are dominant, and NK cell is not activated. Infection of the host cells, especially by some viruses, often leads to an inhibition of class 1 MHC expression, that is to say, the ligands for the inhibitory NK cell receptors are lost. So, the NK cells are released from their normal state of inhibition, and the infected cells are killed. NK cells play several important roles in defense against intracellular microbes. NK cells have granules that contain a protein called perforin, which creates pores in target cell membranes, and enzymes called granzymes, which enter through perforin pores and induce apoptosis of target cells. Interferon secreted by NK cells activates macrophages to destroy phagocytosed microbes. Macrophages are derived from blood monocytes, important effector cells for the elimination of microbes. Macrophages play central roles in innate and adaptive immunity by acting as professional APC. Macrophages express surface receptors such as toll-like receptor, mannose receptor, and scavenge receptor that recognize microbes in the blood and tissues and stimulate the phagocytosis and killing of the microbes. Activated macrophages kill phagocytoid microbes by producing IR, nitric oxide, and enzymes in phagolysosomes. According to the phenotype and secreted cytokines, macrophages are classified into classical activation and alternation activation macrophages. They are M1 and M2-like macrophages. Usually, M1 macrophages promote inflammation, and M2 macrophages inhibit inflammation. Dendritic cells were first found as a population of cells in the mouse spleen by Steven in 1973. DC were named due to a characteristic morphology with striking membranes or spine-like projections, resembling the dendrites or neurons. DC, which are found in abundance in the T-cell areas of lymph nodes and spleen, are the most effective cells for the initial activation of naive T-cells. DC pick up antigens in peripheral tissues when migrate to lymph nodes, where they express high levels of adhesion and co-stimulatory molecules, as well as class 2 MHC molecules, which interact with TCR and CD on helper T cells. Most dendritic cells are thought to arise from adult bone marrow precursors. It is now clear that there are two major populations of dendritic cells that differ in their phenotypic properties and major functions. The two populations of DC are conventional DC, CDC, and plasmacytoid DC, PDC. According to the maturation stages, DC are classified into mature DC and immature DC, and they have different names in different tissues. Regulator TC negatively regulates immune response and maintains immune tolerance. Follicular DC stimulates hypermutation of B cells in germinal center. With maturation, the ability of DC uptaking and processing antigens decreased, while its ability of antigen presentation and stimulating naive T cell activation increases. In addition, mature DC express high level of MHC, co-stimulatory molecules, and cell adhesion molecules, which are required for stimulating naive T cell activation. As the most effective APC, DC initiates naive T cell response, and FDC involves in humoral immunity by helping B cell proliferation and differentiation. DC can regulate other immunocytes by secreting multiple cytokines and chemokines. For example, DC drives Th1 response by secreting interleukin-12. Immature DC play critical roles in both central and peripheral immune tolerance. Adaptive immunocytes refer to T cells and B cells that have specific antigen receptors and are capable of activation, proliferation, and differentiation in response to antigen stimulation to produce a specific immune response. Mature T cells bear important molecules on the cell surface, including TCR CD3 complex and accessory molecules, which include CD4 or CD8 as co-receptors, CD28 and CD2 as co-stimulatory receptor, as well as cytokine receptors. T cells can be classified into several subsets according to the activating stage, TCR type, CD4 or CD8 expression, and T cell functions. According to activating stage, T cells are divided into naive T cells, activated T cells, effector T cells, and memory T cells. According to CD62 ligand expression, memory T cells are divided into TCM and TEM, and ensure distinct homing potentials and effector functions. According to TCR type, T cells are divided into alpha-beta T cell and gamma-delta T cell. Alpha-beta T cells only recognize peptide MHC complex presented by APC and show MHC restriction. However, gamma-delta T cells do not recognize MHC-associated peptide antigens and are not MHC restricted. According to CD4 and CD8 expression, T cells are divided into CD4 positive T cell and CD8 positive T cell. According to functions, T cells are divided into helper T cell, cytotoxic T cell, and regulatory T cell. T-edge cells belong to CD4 positive T cells. TC cells, also called CTL, belong to CD8 positive T cells. Treg cells are usually identified by detecting CD4, CD25, and FOXP3. T-edge cells can be further divided into several subsets according to secreted cytokines. Mature B cells bear functional molecules on their surface, such as BCR complex and accessory molecules, which include CD19, CD21, and CD81, as the B cell co-receptor complex, B7, CD40, and cell adhesion molecules, and cytokine receptors, etc. B cells also act as professional APC. According to CD5 expression, B cells are classified into B1 cell and B2 cell. B1 cells represent a distinct lineage or develops from fetal liver-derived progenitors. The majority of B cells develop in bone marrow, called B2 cells, can further commit to development either into marginal zone B cells or into follicular B cells. Treg cells express CD1D and CD5, performing inhibitory functions by secreting interleukin-10. Lymphocytes continuously move between sites throughout the body through blood and lymphatic vessels. This process is called lymphocyte recirculation, which is critical for the initiation and effector phases of immune responses. Particular subsets of lymphocytes selectively enter some tissues but not others. This process is called lymphocyte homing. Lymphocyte recirculation is very important. Firstly, it enables the limited number of lymphocytes in an individual that are specific for particular foreign antigens to search for that antigen. Secondly, it ensures that particular subsets of lymphocytes are delivered to the particular tissue where they are required for adaptive immune response. Thirdly, fulfill lymphocyte tissues with circulated lymphocytes. Finally, recirculation of memory cells promote secondary immune responses efficiently. Immune molecules are those that are produced by immunocytes or other cells and participate in the immune response. Immune molecules may be secreted or membrane-bound. We will learn immune molecules next time. So, in this part, you should know the composition of immune system. We learned major innate immunocytes, including NK cell, macrophage, and dendritic cell. In addition, we introduced T cell subsets and B cell subsets. Thanks for your listening.